Okay, now let's take a look at some titration curves for amino acids and a few amino acid problems in a couple of different forms. So first we're going to look at the titration curve of glutamic acid. And I have a graph of that already here. The first thing that you always want to do is draw the structure of the amino acid at several different, the points at which they get ionized, or in one case here, isoelectric point, but I'll talk about that as we go through it. So glutamic acid has three pKs, or three ionizable groups in it: the alpha amino group, the alpha carboxyl group, and the carboxyl group on the side chain. So when the pH is 0, which is the first structure I'm going to draw, it's also convention to use a three-letter abbreviation and the charge of that amino acid at that pH. Sorry, got squishy there again. And at a pH, whoops, I'm jumping ahead of myself. At pH of 0, the alpha carboxyl group is protonated, the amino group is protonated, and the side chain is all protonated. So the overall charge is +1. The next time we see an ionization, or the first ionization that is the alpha carboxyl group, and that has a pK of 2.2. And also at this point we hit the isoelectric point, because this amino acid now will be uncharged. The negative charge of that carboxyl group is going to cancel out the positive charge on the alpha amino group. But we still haven't hit the point where we will be ionizing that side chain. So pK1, this is the pK of that alpha carboxyl group. So at 2.2, this will ionize, and we will have this pink region. We have a buffer region in the flat part of that. We also have a buffer region here, and we would have one up here as well. But the isoelectric point, again, is the pH at which that amino acid has an overall net charge of 0. Now, the next structure we're going to draw is when the side chain ionizes. Okay? And when that happens, we're going to have an overall net charge of -1. So we have the ionization of the R group. So now we have two -1s and a +1, so we have an overall net charge. And this corresponds to at this region. Okay, we've completely ionized. We're in equilibrium here. And then finally, when we get up to 9.4, this was 4.3, sorry, pK of that side chain, 4.3. We will have deprotonated, the alpha amino group. And, excuse me, our overall charge now will be a -2. At this state. So you need to be able to draw these curves and or label them, identify where the PKs are occurring, where the isoelectric point is. So let's take a look now at the titration curve of lysine. And I've got the graph here, but let's first draw the representations of the different charge states. And at 0, we have, and just to conserve a little space here, I think I'm going to start drawing these with that group up, just so I'm not running out of room. I'm also going to consolidate those methylene groups. Oops. Oh no, that was right. I'm so used to having that there. Okay, so when the pH equals 0, lysine has a plus 2 charge. Both the side chain amino group and the alpha amino group are charged or protonated. So the next ionization, or the first ionization I should say, is going to occur on that alpha carboxyl group. I'm not used to drawing these this way, so bear with me. So we've got an offset of charge here. We still have the, we have 4 methylene groups, but we still have that side chain amino group charged. So at this point, the lysine has a +1 charge state overall. Okay. And this pK, of course, is going to be at 2.2. So that means, let's say, there's about 2.2. I'm just gonna, we won't have another ionization until we hit the, I'm just gonna go that far for now. So this is PK1, and we have some buffering going on here in that buffer region. So we don't get another ionization or deprotonization until we hit the PK for the alpha amino group, and that is at 9.4. And at 9.4, this will get deprotonated. But our alpha amino group still carries a charge. So now lysine is at its isoelectric point with an overall charge state of 0. And let's say this is about, I don't know, 9.4 here. So this is the PI, and I should flatten this out a little more in between here, and this is of course not to scale. We have our second PK, and then we'll go up a little bit more, where we'll flatten out again, and the last charge state for lysine is a -1. So we're also going to deprotonate the amino group in the side chain. So we will just have the -1 on the, for alpha carboxyl group, so a -1 here. And that PK again is at 10.5. So I've run into the chain here, but we'll call this region the PK3. and that is the titration curve for lysine. Okay, now let's look at some amino acid problems that you're going to be expected to be able to work. We have two that involve using the Henderson-Hasselbalch equation, and they involve also the amount of dissociation or ionization that has occurred in the amino acid, for example. So the first question asks for the pH when 1/4 of an alpha carboxyl group is dissociated, okay? So the pK we know is 2.2, and we're just going to use the Henderson-Hasselbalch equation. So pKa is 2.2, and what we need then to do is add to that the log of COO minus over the concentration of the weak acid or that carboxyl group that is unionized. So if 1/4 is ionized, then 3/4 are not, correct? So we can use those fractions in our ratio. And if 1/4 is ionized, that is the concentration, so to speak, of the conjugate base. 3/4 are not. These cancel, so we get a ratio of 1/3. So our pH equals 2.2 plus the log of 1/3, and that gives us 1.72 for our pH. And we have another one of these, and in this one, the alpha carbonyl of glutamic acid is used, or we're looking at, is 2/3 dissociated or ionized. So just in general, again, we use the Henderson-Hasselbalch equation, and we're looking at, again, the concentration of conjugate base of that alpha carboxyl group divided by the undissociated, okay? So same rationale, if 2/3 is dissociated, 1/3 is not. So we've got a pH, and I'm sorry, this was an AMA, this was an alpha, this was gamma. I drew it gamma, but I said alpha, sorry about that. So this is the carboxyl group in the side chain, and that has a pK of 4.3. And we just said we had 2/3 dissociated, meaning 1/3 is not dissociated. Again, those cancel. So we're taking the log of 2, and pH here then equals 4.60. So you can expect to see a calculation like this possibly on your first exam. So some other types of amino acid problems you might see on an exam, and actually I guarantee you will see one of these, and likely you will, it will take the form of not this one, but one coming up, and I'll point that out in a moment. But it's fair game to ask you to draw some type of peptide. This is going to be a dipeptide, so we have 2 amino acid residues, and I always, unless otherwise specified, want to see those at physiological pH, or 7.4. Okay. So we're going to draw a peptide, dipeptide, so just 2 amino acids. The N-terminal peptide residue is aspartic acid or aspartate, followed by histidine. So we're going to have that alpha amino group that is protonated. Remember, we've got an amide bond here linking these 2 residues together. And at physiological pH, that carboxyl group is ionized. It has a pK of 3.9. Okay. And our next amino acid is histidine. Now this has a free carboxyl group at the end, so it's charged. Ooh, I've got a rumbly tummy here today. And keep in mind again, the pK of this R group is 6.0, so it is mostly deprotonated at physiological pH. Okay. So that is the way we would show it. Now there are a couple, actually I'm sorry, one other type of calculation that you should be able to do, and this is a minor thing. What if you're asked to calculate the number of possible peptides? That contain one of each of these amino acids, or one residue of each. Alanine, glycine, histidine, lysine, val. And the identities of these doesn't really matter. This would be the same for any group or given group of 5 different amino acids that have only appear once. So the number of possible arrangements of these 5 amino acid residues is 5 times 4. So, for example, the first residue could be any one of the 5, and the second could be any one of the 4, and so forth. Whoops, I'm sorry, that should be a 2, times 1. So 120 different sequences. So that's what makes the variability of our replication system going from DNA to RNA to protein so amazing, because out of very small numbers of amino acids, we can get enormous numbers of sequences, essentially. Okay. So let's go back to some types of drawing amino acid problems you might see. Like I could ask you to determine the net charge. In this example, the predominant form of aspartic acid. Or ASP at, in this case, A is a pH of 1. So when the pH is 1, the only charge carried is by that alpha amino group. So overall net charge at pH of 1 is going to be a +1. And the next pH you're asked to calculate or draw the structure for is at a pH of 6. And so by that time, we've hit the pK of both the alpha carboxyl group and the side chain group. So now we're going to have a net charge of -1. Okay, last one of this set, and then we'll take a look at the reactions of amino acids. You've already seen these on slides, but I would like you to be able to draw them as well. So the last in this group that's in your packet is you are asked to give the overall net charge of a tripeptide at physiological pH. Okay, so that means you've got to draw it. You need to show me where the charges appear and give the overall net charge. And you are given the tripeptide ASP, lyse, ASN. So aspartic acid or aspartate, lysine, and asparagine. Again, this is the N term. This is the C term of this peptide, or tripeptide. So the N term will be protonated, and at physiological pH, the side chain of aspartic acid will have ionized. Next, we have lysine with three or four methylene groups, and its amino group will be protonated. And lastly, asparagine. Its alpha carboxyl group will be deprotonated. So we have 2 positive charges and 2 negative charges. So the overall net charge of this tripeptide is 0. So next we're going to look at the reaction of an amino acid with Edman reagent. This is what's used in the process called Edman degradation. And this identifies or analyzes the M-terminal amino acid. Amino acids. Goodness. Can't spell today. Amino. Okay. So this reagent is going to combine with a free lysine that occurs at the N-terminus of a protein. And then that residue can be excised from the polypeptide chain and isolated as a PTH derivative. And then that can be analyzed and the amino acid can be identified. If we have a polypeptide of about 100 to 200 residues long, we might be able to identify the first 50 amino acids. Okay, so it does have limitations, and we will look at other techniques that will allow us, or do allow us, to get longer sequences in polypeptides, or more sequence in polypeptides. I think I mentioned this weekend class at our meeting that the efficiency of this method with larger proteins is far less. So with a typical amino acid that is about 2,000 residues long, we might get the first 10 to 20 terminal amino acids identified. But let's just we're not going to do all the steps, but I want you to be able to recognize, and you don't need to draw this process, but I want you to be able to recognize the amino acid when it is in that PTH derivative. So we've got our phenyl isothiocyanate, which is the reagent. So that's its structure. And we have our peptide chain, and we're just going to use generic R groups here. So here's an R prime, and this goes on and on and on. But this is the alpha amino group, and it has been treated with an alkaline reagent. So the amino terminus is deprotonated. Well, actually occurred before this, but and then we get this thylosine derivative. So here's the portion that belongs to the phenyl isothiocyanate. Here we go. There's our R group, H, and the bond here. And then we treat that with a weak aqueous acidic solution. And we get the PTH derivative. So we have that. This is our derivative. So that's our PTH amino acid. So let's take a look at the formation of an amide bond. Okay. So this is what occurs when we basically polymerize amino acids. Again, I'm just going to use a generic R group for each of these. And I'm going to draw the deprotonated alpha carboxyl group like this. And for the upcoming or incoming amino acid, I'm also going to show that so you can see all the hydrogens here. So what happens is we are going to have a reaction between that oxygen and two hydrogens. We're going to release a molecule of water. whoops, H2O and form that amide bond. Whoops, sorry about that. So in general, we could call this a condensation reaction. We're condensing two smaller molecules into a larger molecule, in this case a dipeptide. You could also call it dehydration, as we're releasing a molecule of water. And the other reaction we want to look at is disulfide bond formation. This is also, I've mentioned that hydrogen bonding is critical to protein folding and structure, and again we'll talk about that when we talk about proteins. But this disulfide bond is also a stabilizing factor. So this is going to occur, and I'm just going to draw a portion. Oops, sorry about that. Make that a C. Of a peptide chain, and this is going to go on. So this is a cysteine residue. And it can react with another cysteine residue within the same polypeptide chain to form the disulfide bond. Okay. And we are going to need half a mole of O2 and these two hydrogens. They will react and form a molecule of water. And the end result then is the linkage between these two polypeptide, or between these two amino acids in the polypeptide chain. And this is our disulfide bond, or you may hear it called, when we talk about proteins, the disulfide bridge. Actually, that should have just been a continuation there, because we've got more amino acids in this chain. So that's it for amino acid types of reactions. Now we're going to look at amino acid analysis problems. Okay, now for my favorite type of amino acid problem. So the analysis of short oligopeptides. We did one this week when we met on Wednesday. So I have given you two in this packet, and I believe I had suggested practice problems that include a few more from your text. I encourage you to work those, especially if you're having trouble. Again, keep in mind that on an exam, I will give you the specificity of any enzyme that is used in the treatment of these peptides. So in the first one, and I'm going to switch to just plain paper to do the actual work, but we're told that we have an oligopeptide with 7 residues, so 7 amino acids. And they give us the identification of those amino acids. However, there are only 6. So that tells us that at least one of them occurs twice. So in number 1, we are given the identity or the composition of this 7-residue oligopeptide, or we could call it more specifically a heptapeptide. Sorry, here we go. But one occurs 2 times. Okay, so that's the information we have from the first step. And I'm going to draw my 7 lines. And keep in mind that each treatment or each step is done on the full polypeptide unless we're told otherwise. Okay? So in 2, we're given 4 different sets of treatments and observations. So we'll take a look at A first, which is digestion with trypsin. And the observation is no effect. Nothing happened. Okay. And this cleaves or cuts the oligopeptide or oligopeptides C-term or C-terminal of arginine or lysine. Okay? Do we have an arg or lys in this polypeptide or oligopeptide? Yes, we do. We have 1. And if trypsin did not have an effect, that means lysine must occupy the last position because it was not released. So from 2A, we can put lys at the C-term position. Okay? So that's from A. In B, we are told that, or we're shown, the treatment with phenylisothiocyanate or Edman reagent. PTHC. And we have the release of a derivative, PTH derivative, that contains alanine. So we get phenylalanine. Didn't look at the whole word. Or my structure. So taking a look at our sheet again, this is right here, we've got the alpha carbon. This is the alpha amino group here, and this is phenylalanine. Okay? So that means we can place phenylalanine at the end term of our amino acid. Or peptide. In C, we are treating this 7-residue peptide briefly with chymotrypsin. Okay. And this cleaves strongly, C-term, of phenylalanine, tyrosine, or tryptophan. Strongly. And for future reference, it does cleave blue weakly, but we're only briefly treating this so that cleavage should not occur. And with that treatment, we get a dipeptide and a tetrapeptide. So let's think about this. I'm not sure we're given all the information of these, or all the observations in this particular treatment, because what else would we only have 6 amino acids here. What else would be released? We're also given the amino acid composition of this tetrapeptide: leucine, lysine, and methionine. So, if we treat this with chymotrypsin, we would expect a cleavage of the N-term phenylalanine. So we get that from C, and we're not told about that. And we get a dipeptide and a tetrapeptide. That tetrapeptide contains lysine. We have a lysine at the C term of our amino acid. So that tells me that the other cleavage occurs here. And this, since we have tyrosine, I'm going to place here. Okay. So I think that's all the information we can glean here. The tetrapeptide did not contain phenylalanine or tyrosine, so it has to come from a C-term cleavage at position 3, which is what we have here. Okay? And in D, we treat with cyanogen bromide. Or CNBR, to shorten that. Here we get a dipeptide, a tetrapeptide, and a free lysine. Cyanogen bromide cleaves C-term of methionine. Or MET. Okay? So, since we get 2 peptides and a free lysine, that means there must be 2 METs or 2 methionines. Okay? And one of those has to be in the 6th position. Otherwise, we would not get a free lysine. So here's methionine. We know we've got a cleavage here, and that comes from D. So we still have 3 unassigned residues. Another MET, second one, leucine, and aspartic acid. However, to get a tetrapeptide, the second MET has to be in the 4th position. And this will give us a tetrapeptide, a dipeptide, and a free lysine. So we also get this information from D. So now we can go back and take a look at leucine and aspartic acid. We were told in C that leucine occurs in the tetrapeptide with lysine and methionine. So leucine must be here. And then by default, aspartate or aspartic acid must be in the second position. Okay, we've got another one. I hope you're following this just fine. If not, send me an email or ask about it when we meet again next Wednesday. We did walk through one of these in class, but let's try another one. And this will conclude the amino acid segment for this first exam. So the second one I have here is an octopeptide. So we have a total of 8 amino acids. The composition is given to us. Again, this is not sequence, but the identity of the amino acids that are present. 2-alanine, an arginine, 2-tyrosine, valine, aspartate or aspartic acid, and methionine. The first observation is partial acid hydrolysis. And this gives us a dipeptide. Okay. This what are the identities of these two amino acids? Alanine and valine. Okay. So, we have an octopeptide, which again is 8 residues. And we know we have 2 alanines, an arg, 2 tyrosines, valine, aspartate, and methionine. Also, we're told that the ammonium ion was detected, and we'll come back to that later. Okay? Partial acid hydrolysis in 2 gives a dipeptide, alanine, valine. Okay. In B, we're treating with chymotrypsin. Again, this cleaves C-terminal to tyrosine, tryptophan, and phenylalanine, and this again is strongly. Lou and Matt Weekly, I don't think I included Lou before. And from this treatment, we get 2 tetrapeptides. And each of these contain 1 alanine. Okay. So in order, what we can glean from this, I guess we should start our little lines here. In order to get 2 tetrapeptides, we can identify the positions of our tyrosines. Okay? So if 2 of those were internal, we would get 3 fragments. But we only get 2 fragments. So that means one of those tyrosines has to be C-term, and the other one has to be in the fourth position. Okay. So we've identified the position of our tyrosines. In C, we treat with trypsin. And so we're treating tetrapeptides here. One of the tetrapeptides is going to, or it gives us 2 dipeptides. And again, trypsin cleaves C-term of arg or lyse. We do have an arginine. Okay. So this is what our tetrapeptide looks like from this information. Let's take a look at D, because we still don't know exactly where this is yet, okay? In D, we treat with cyanogen bromide. And this gives a tripeptide and a free tyrosine. Okay. So we're treating the same tetrapeptide as we are in B. Okay. So this is the same tetrapeptide as we treated in B. I'm sorry, not B, C. So, what does that tell us? Tells us that we have methionine here and tyrosine here. Okay. Excuse me, one moment. Okay, so that tells me that we have arginine and methionine here. And, whoa, where do I want to go? Back to B, 2 tetrapeptides contain alanine. Okay. So we're going to find one of those alanines here. So let's take a look at the last treatment, and this was with Edmond reagent. So that, again, is going to identify the N-term amino acid. And that was followed by acid hydrolysis, which released that ammonium, or the alpha amino group. So that tells me, and the derivative, PTH derivative, was aspartic acid. So aspartate is in the first position. This is from E. Sorry, I haven't been continuing to label these. We got tyrosine from B, step B, methionine from D, arginine from C. This is by default because we know that both of the tetrapeptides contained alanine. And in A, we found the order of this dipeptide. So this information was found in A, or this observation was from A. So now we have the complete sequence, or the correct order, of the amino acids in that oligopeptide, 8 residues. All right, thanks. Try some of these on your own.